Understanding the pathophysiology of and the immune response to infectious disease is vital for discovering effective treatments. The following two case studies address several problem-oriented metabolomics applications for SARS-CoV-2 research. In this study, global metabolomics were used to identify lipid metabolites which changed in alveolar type 2 epithelial cells during influenza infection. The method here was isolation of AT2 cells from mice that were infected with H1N1 influenza to induce acute respiratory distress syndrome. So like influenza, severe COVID-19 infections can lead to respiratory failure from acute respiratory distress syndrome. And this respiratory failure from ARDS is associated with increased mortality. Global metabolomics were performed on these isolated AT2 cells. This is important because these are the primary site of infection for influenza virus. And acute respiratory distress syndrome in influenza infection is associated with significant alterations in AT2 functions. So the investigators were wondering if there would be metabolomic changes that were also associated with ARDS in these cells. So here you can see that infection resulted in significantly reduced levels of several surfactant phospholipids, including phosphatidylcholine and phosphatidylethanolamine, as well as increases in cholesterol. Collectively, these can result in alterations in surfactant. And when surfactant is changed, those can lead to increased surface tension in the cellular membrane, changing the functionality of the alveoli. There were also decreased CDP-D choline and CDP-ethanolamine in these cells, and increased phosphoethanolamine and diacylglycerols. These can be associated with changes in phospholipid synthesis. All of these changes result in significant alterations in AT2 function, which can lead to acute respiratory distress syndrome. So in this study, the investigators were able to identify potentially actionable alterations in the lipid metabolome that is associated with acute respiratory distress syndrome during an upper respiratory infection. In this study, global metabolomics were utilized to identify predictors of disease severity and the need for ventilation during respiratory infection and bronchiolitis in infants. The severity of bronchiolitis varies significantly among infants, and it's thought that the interactions among the respiratory viruses that infect the infants, the upper airway microbiome, and the host immune system could impact the severity of the disease. So utilizing the Mark 35 cohort of just over 1,000 infants who had been hospitalized for bronchiolitis, the investigators obtained nasopharyngeal aspirate samples to have both metabolomics and metagenomics run to identify markers of the severity of infection and the need for positive pressure ventilation. So positive pressure ventilators are increasingly in demand to treat the most severely infected of COVID-19. And so here, another respiratory infection may serve as a model to help understand the interplay of the various factors that would cause the need for this intervention. So using the NP samples, metabolomic analysis identified biochemical markers associated with a higher risk for severe infection, which are shown in the red, and a lower risk for severe infection, which are identified in blue. So you'll see that some of the biochemicals that were associated with the more severe infection include sphingomyelins and sphingosine, while things like inflammatory lipids were negatively associated with disease severity. Interestingly, a series of microbiome-associated metabolites were also identified in this analysis. These could also be used to predict the need for positive pressure ventilation in the infants. So further, the metabolomics and metagenomic data were integrated to correlate the members of the nasopharyngeal microbiota with the metabolites associated for positive pressure ventilation. So what you can see in A and B panels are that Streptococcus is pneumoniae abundance is positively correlated with the metabolites that were most highly correlated with disease severity. So here, metabolomics of the samples taken from the site of infection were able to identify those who were at risk for the most severe infection, and they can be integrated with other omics to make predictions for the need for positive pressure ventilation as an intervention, and disease severity.